Hi, this is the Combined Science Trilogy Higher Paper, Paper 2, Biology for Friday the 7th, June 2024. Okay, it's the AQA version. Right, so question number 1. Limpets are small animals with shells. Limpets attached to rocks on the seashore. That's a nice little line of them there, look at that. Figure 1 shows limpets on rocks. Okay, limpets eat algae. Okay, limpets are prey for crabs. Okay, give the food chain for algae, crabs, and limpets. Okay, so put them in the wrong order. So what you need to do is you need to say algae is eaten by limpets, which is eaten by crabs. Okay, so don't forget the arrow means eaten by. Students estimated the population of limpets on the seashore. Okay, that sounds like a nightmare, right? The students were given a square quadrat. Okay, so there we go. So it's 50 centimeters by 50 centimeters, right? Calculate the area of the quadrat in meters squared. Right, so first thing we need to convert both sides into meters squared. So it's 0.5 meters squared multiplied by 0.5 meters squared equals 0.25. Okay, make sure then you write your answer in the bottom there, which is 0.25 meters squared. It's quite straightforward, that one. The total area of the seashore was 1,800 meters squared. The students sampled 2% of the total area of the seashore. Calculate the number of times students needed to use the quadrat for a 2% sample. Okay, use your answer from question 1.2. So we've got 0.25 meters squared as our quadrat. Right, so first thing we need to do is we need to work out what 2% of 1,800 meters squared is. So I'm going to do 1,800 multiplied by 2 over 100 equals 36 meters squared. Okay, so that is then 2%. So that refers to 2% of the total area of the seashore. Now what we've got to do is if we've got 36 meters squared, how many times do we use the quadrat? So therefore, if we've got 36 meters squared, right, we divide that by 0.25, okay, which will then give us 144 quadrats. Okay, you could also do it in a similar sort of way. I'll just do it in a different color. If you've got 36 meters squared, right, and if each quadrat is 1/4 of a meter squared, you could then do 36 multiplied by 4, which will then give you the same answer. Okay, don't forget, write your answer on the answer line. Question number 1.4. Explain why throwing a quadrat is not a random method to estimate the size of a population. Do not refer to safety. Why would anybody ever refer to safety? Throwing a frisbee across a field. Okay, so what it could be biased, right? Because if you think about it, if you're looking for the number of daisies or whatever it could well be on a field, and you see a patch of daisies, the natural thing to do is to try and throw the quadrat towards the daisies, and that would then be biased. You can also limit how far you can throw. Okay, so if you're on the beach, right, and you're looking for the limpets, right, and it's 1,800 meters squared, that's a fairly big area. So what you've got to do is you've got to be able to then cover the entire area. So it could be biased because you might aim for the limpets, and it could also be difficult to throw it to a farthest end. Question number 1.5. Okay, describe one method the students could use to plan where the quadrat would be randomly placed each time. Okay, I like this, right? So what you have is you have tape measure on two sides. Okay, so let's imagine it as a rectangle. So what you'll do is you measure, in blue, you measure that side, and then you measure that side. We'll put a tape measure down. Okay, that's two sides of the seashore. Okay, because what you've got to do, you've got to make sure you keep referring back to where it is. Then give coordinates or label the axes, coordinates axes or the sides. So what I would do is I'd tend to go, let's say it's 20 across there, and then I'd probably go to A, B, C, D, E, F, G to Z, or whatever letter it is. Okay. Then what you've done is you've then turned it into a grid. And then once you've actually got it in a grid, to get it as random as you possibly can, use a number generator. Okay, so then when you use a number generator, it might come up with 2p. And if it comes up with 2p, you then know exactly where you're going to put it, and it's random. 1.6. Suggest one hazard the student should be aware of when collecting data on the seashore. Do not refer to throwing quadrats, because that wouldn't be dangerous, wouldn't it? I mean, students throwing quadrats. Okay, right, so if you think about it, you're by the sea, so there is a water hazard. Okay, you're also by the sea, so therefore there could be a tide hazard. Okay, you're on limpets or looking for limpets, so there's going to be rocks all over the place. There could be rubbish like glass. Okay, there could be sewage. Okay, and there could be animals. Okay, so there could be other animals that might cause some sort of hazard in itself. I'm not quite sure what they could be, right? But you know, and then what you do to do then is you need to say a hazard is the tide, because it could come in when you're not expecting it. 1.7. Populations of limpets are monitored to assess the impact of pollution of water. Okay, that makes sense. Suggest one type of pollution in water that may affect the population of limpets. Okay, I'm going to write down it properly this time. I should have written it down better in the last one. A pollution could be fertilizers, or it could be sewage, mentioned that one previously, or it could be toxic chemicals. Okay, so when you're actually writing your answers down on these, make sure that you're not doing like a one-word answer. So on 1.6, where it says suggest a hazard, don't just write around rocks. You're going to write down rocks could be sharp, you could hurt yourself if you fall on them. Okay, that's justifying it a little bit better. Question number 2, potatoes. Okay, potatoes are a food crop. Potato plants are classified as eukaryota. What type of classification group is eukaryota? They are a domain. Okay, it's the biggest group. Okay. Question number 2.2. Potato plants can reproduce by asexual reproduction. Okay, little runners that kind of run underneath the ground. Which statement is true for asexual reproduction? Meiosis? No, because that is your sperm and your egg. Okay, that's producing two gametes. Offspring are genetically identical. Correct. Okay, so if you think a potato plant goes underneath the ground, a little runner, and it pops up somewhere, it's the same plant. Okay, pollen and egg cells are produced. No. 2.3. Flowers of potato plants contain gametes. Gametes is then your kind of your equivalent to the sperm and your eggs. Okay, so that would then be your pollen. Okay, how is a gamete different from other cells in a potato plant? Okay, so what you got to think, it's gone through meiosis. A gamete contains 1/4. Why on earth would it contain a quarter? A gamete contains half of the number of chromosomes. Correct. A gamete contains double the number of chromosomes. Not a chance. Okay, so you've got to remember meiosis. What you do is you start with one individual cell. Okay, and as it goes down, eventually what you're going to do at the end, you will then have 4 non-identical daughter cells that have got half the number of chromosomes. That's meiosis. 2.4. Plants of the same genus as potatoes have been studied. Okay, so you've got genus and you've got species. They're the two lowest ones kind of on the classification system. Describe one way a new plant species could be identified as being the same genus as potato. Okay, so that means it's very similar. All right, so it could be a new plant has a similar structure as potato. Okay, We could also have a new plant. I'm being lazy here now, don't you do this. Could have similar characteristics. You know, like the sort of same short-shaped leaf, same sort of flowers as potato. A new plant could have similar DNA as a potato. A new plant could have similar biochemistry. Okay, that's probably the hardest one to kind of think of, right? But the best ones on there, it could have similar characteristics, like the same shaped leaf, same height, same type of flower when it flowers. Oh, big question. 2.5, right? Scientists have collected and stored seeds from species of the same genus as potatoes. Okay. In the future, these seeds may be used to genetically modify a potato plant. Fair enough. Genetically modified, so your GM, modified plants, potato plants, could help supply food to the human population as the climate changes. So we're talking about getting food for humans because climate is changing. Explain. So then we need to think about why genetically modification of a crop plant may be important for human population to survive climate change. Right, so what we've got to do there, we've got to kind of break it up into individual bits or sections. First thing, let's talk about what climate change is. Climate change. Climate change. All right, so this is then where what you're going to do is you're going to get change in weather patterns. Okay, you're also going to get potential flooding. You're going to get potential drought. You're going to get farmland could be affected. Oh, got it right. Okay, current crops may not survive. Not survive. Okay, so what you got there is you've got climate change and current crops, right? So basically, the crops that we've now got, they might not be able to survive as the climate in the UK or wherever it is in the world changes. Okay, because it could become drier, it could become wetter, right? And they may not be adapted for it. So then what we can do is we can then do GM crops and the benefits. So really what I'm trying to do here is I'm trying to break it up into sections. And we're talking about climate change, which is the issue. Now we're going to talk about genetic modification, right? So the benefits. You can make crops that will grow in a wider range of conditions. Okay, so your wheat might be able to grow in drier conditions or maybe in wetter conditions, depending on where it needs to be. Crops can be more resistant to pests plus diseases. Okay, loads of crops all over the world just get destroyed by insects and diseases. Okay, so then if you then eradicate that, then what you're going to do then, you're going to have more food. You also could get an increased yield, right, which is exactly what's happening now. So your corn on the cob might have only been small in the past, but now it's a lot bigger. So you get an increased yield, which means you can feed more people. Explain. So if you have an increasing population, it can then be fed using smaller areas of farmland. Okay, because we're going to be building houses all over the place. Certain places might just flood all the time and have drought all the time. So the amount of farmland is going to decrease. So the GM crops is then going to allow us to produce more food so that then we can feed the population. Oh, I love family trees. Okay, AKU is a genetic disorder. Okay, I have no idea what it is. Okay, sounds very, very exciting. All right. Figure 3 shows the inheritance of AKU in one family. So let's have a look. So a male who has AKU. So that dude there is a male who has AKU. That's a female that has AKU. Male that doesn't have it. Female that doesn't have it. Describe how figure 3 shows that the allele for AKU is recessive. Right, if we look at A and B, mum and dad, okay, they have produced a child that has AKU. Okay, now if AKU is dominant, both A and B would also have it. Okay, so therefore, parents of a child with AKU do not have AKU. Okay, they haven't got it. So what they must be recessive. Okay, so which person is definitely heterozygous? Okay, so we look at C, right? We don't know if C is heterozygous, right? Well, they're probably definitely not heterozygous because D must have little a and little a, okay, which is then the two recessive alleles, right? And therefore, H and I will be heterozygous, okay, but that isn't an option, so it's not that one. D is definitely not because D has got it, so they will then be homozygous recessive. E, right, they haven't got it, right, but we can't tell from A and B what they've got. G is giving it to the kids, so therefore F must be heterozygous, but that isn't an option, and G must be heterozygous because K and L have got it. So G is the option. A female who has AKU and a male who is heterozygous AKU, right, plan to have a child. Fabulous. Good on them. All right. Determine the probability that the child will have AKU. Complete figure 4. Okay, so figure 4 is our Punnett square. Identify the phenotype of each offspring genotype. Okay, so using the symbols. So A is the dominant allele. That means they haven't got it. A is the recessive allele, which means they have got it. Okay, that is the one. So a female, let's look at the top one here, who has AKU. So therefore, if they've got AKU, it must be A and A. And a male is heterozygous, so that means A and A are having a child. Right, what's the question? Probability that the child would have AKU. Right, so let's do our Punnett square. So we've got, let's change colour, let's be exciting. A. Sounds all very good, doesn't it? All right, so A is heterozygous, and so they will not have it. AA is homozygous recessive, so they will have it. So the probability that the child will have AKU is 0.5, 50%, or 50%, a half. Okay, I would go for the decimal each individual time. 3.4. A mutation is a change in a gene. People have AKU have a mutation that causes the production of a non-functioning enzyme. Okay, so what you've got is an enzyme that doesn't do its job. All right, so this could be an enzyme, or draw it on Pac-Man. Okay, so that's an enzyme, and what you've got is you've got this active site in the middle. Enzymes are proteins. Suggest how a mutation can result in the production of a non-functioning enzyme. Right, so what this Pac-Man here is, this protein, this enzyme, right, what it is, it's just a big long chain of amino acids. Okay, and they're all in a very, very specific order. If you've then got an incorrect sequence of amino acids, the enzyme will change shape. Okay, so let's say this little amino acid there changes, and then what it does, it then changes the entire shape because of the way they're linked together, right? Then the active site will change shape, and then the enzyme then won't work. 3.5. Some people have in vitro fertilization, in glass fertilization, treatment to increase the chance of becoming pregnant. Describe how process of IVF can result in pregnancy. Fabulous. Okay, so let's just do it in sections, right? 4 marks, great question. FSH and LH are given to the female. Okay, so FSH is follicle stimulating hormone, so the follicles in the ovary are then stimulated to become mature. LH prepares them and causes ovulation. So therefore you've now got stimulated mature eggs. Okay, part number 2. Eggs are then collected. Okay, so what they do is they get a massive great big long needle, shove it into an ovary, suck out the eggs, okay, then go for the other ovary, suck out the eggs from that, and then you've got them. Fertilize the eggs using sperms. Okay, and then they will start to replicate using mitosis. Final one. 1 or 2, usually 2, are reinserted into the uterus. Okay, so they usually put 2 in, right? Because if they put 2 in, then there's more chance of them getting pregnant. However, there is more chance of getting twins. 3.6. Embryos can be screened to detect inherited disorders. Okay, give two arguments against embryo screening, because obviously a good thing about embryo screening is you can find out if there is some significant issues with the embryo. Do not refer to religion, right? So there is a risk to the embryo. Okay, imagine if you're taking cells from the embryo and the embryo is tiny, there is a risk to it. It could lead to a termination. Okay, hence the risk to the embryo. It could cause damage, right? It could also lead to the embryo coming out of the lady, which is then a termination, so then the lady is not pregnant anymore. It is stressful for the mother and the baby, or the embryo rather, and it will also cost to have the procedure. Procedure. Question number 4. Rats. Okay, rat population can increase rapidly. I bet they can. When rat populations are large, there is competition between rats. Okay, fair enough. When rats compete, the basal metabolic rate. Okay, so metabolism is all the chemical reactions within the body. So when they compete, their metabolism increases within the rat. It's controlled in the same way as humans. Okay, they're being clever here. Okay, so we're talking about metabolism in rats, but they're telling us it's the same way in humans. How is it increased? Thyroxine from the thyroid gland is secreted. Okay, so what I've done then is I've got thyroxine, which is the hormone from the thyroid gland, and then I've got the thyroid, right, is also in there. You could also say TSH, thyroid stimulating hormone, could be produced from the pituitary gland. 4.2. Okay, so 4.2, the size of a rat population can increase quickly. One female rat and one male, 20 kids in 2 months. That's just shocking. Warfarin is a poison that has been used to control rat populations. Rat populations can become resistant to warfarin, right, by the process of evolution. Okay, a population of rats was given warfarin for 4 months. So 0, 1, 2, 3, 4. So they were given it, right, and the percentage, or table 1, shows information about resistance to warfarin in the rat population for a year after warfarin was given. Okay, so we got it for 4 months, Resistance gets up to 81% within those 4 months. And after 6 months, 99% of rats are resistant to warfarin. That's shocking. But then what you can do, then you can see it goes down. It starts to go down quite slowly, but it goes down. Right, rats with a resistance to warfarin have a smaller mean mass. So the resistant rats are smaller. Okay, so there's big rat. Okay, and there's the resistant rat. Okay, they're probably not quite as dramatically different as that. Smaller rats are often at a disadvantage when competing against larger rats. Okay, obviously, so the big rat is going to get the food better than the little rat. Explain the trends in data when warfarin was given and no longer given. Okay, so you can see there exactly the way it's splitting it up. Right, so let's go for the trends first. So trends. Okay, there's loads of different trends you can see. The percentage population with resistance increases for 6 months. Okay, can't get much higher than 99% really. Then it decreases after warfarin stopped, but slower. Okay, so what I'm doing there is I'm just describing. Okay, explain it. Okay, because now what I do, I've got to explain it. So, some rats have the resistant allele due to a mutation. Okay, now what we've got to do now is we've got to think about the question again. Being given, which is increase, and then no longer given. So I'm going to do increase first. So the rats with the allele, so with allele, reproduce, passing on the allele. Okay, so those resistant ones, okay, they will survive the onslaught of warfarin, right? And then what they do is they then reproduce, passing on the allele. So therefore, the population is going to increase. And what it's going to do, it's going to go up several generations in 6 months. Okay, so you're producing a lot of little rats. Okay, then after that, it then decreases. Why does it decrease? It decreases when warfarin not given because, I'm going to have to start over here, because non-resistant rats, that looks like cats, but it's not, rats out compete these small rats. Okay, so those ones at the left that didn't die, all right, that are not resistant, right, they start replicating again, or reproducing again once the warfarin's then stopped. And because they're bigger and they're stronger, what they will then do is they will then start to win the population battle again. Okay, question number 5. Okay, otters are mammals that live in river ecosystems. Define the term ecosystem. Okay, definitions. So it is ecosystem is the interaction or interaction of organisms and non-living parts in their environment. God, all right, it's terrible, that one. 5.2. Otters are an important species for the stability of river community. Fair enough. Describe two ways, two, animal species may be important for the stability of the whole community. So it doesn't necessarily mean just otters, but any animal species. Right, so we could have, let's change colour, I like that colour anymore. Okay, the animal may be a predator controlling prey population. It may be a prey which feeds the predators. It could be maybe a primary consumer, which is then controlling plants. It could disperse seeds. So the animal eats something that's got seeds in it and it'll disperse them around the area. Okay, so that other animals, and if you take one of them away, then the whole kind of ecosystem in that area then changes. Oh, that's a lot of words. Okay, 5.3, sewage was accidentally added to a river. What? The sewage moved from the river water and affected the number of bacteria in the water. Nice, because the bacteria are eating it while they're living off the actual sewage. The concentration of dissolved oxygen in the water decreased. Okay, that's then because the bacteria are then using the oxygen in the water, and then the overall percentage of oxygen in the water is going to decrease. Samples of river water were analysed at different distances from where the sewage was added. Okay, so sewage added at 0, number of bacteria per 1,000 millimeters. Number of bacteria in 1,000 millimeters cubed was 4. Concentration of dissolved oxygen was 5.4. So what we can do is you can see in kilometers as it goes away, the number of bacteria increases and the concentration of oxygen decreases. 5.3. The number of bacteria at 5 kilometers was greater than the number of bacteria where the sewage was added. Calculate how many times greater. Quick sum. Okay, so if it starts at 216, which is 5 kilometers away, and at 0 it was 4, 216 divided by 4 equals 54, which means there was a 54 times greater number of bacteria 5 kilometers away. 5.4. A student concluded the number of bacteria is proportional to the concentration of dissolved oxygen. Explain why the student's conclusion is not correct. Right, well basically they're getting it completely wrong, aren't they? As the number of bacteria increases, the oxygen concentration decreases. Okay, so it's opposite to what they're saying. And that is then a negative correlation. Otters. Otters live in water and on the land, and they eat mainly fish. The concentration of dissolved oxygen has decreased in a river where otters live. Okay, well I guess if they live on land and water, it's probably not going to affect them. Explain how the decrease in concentration of dissolved oxygen in the river water will affect the population of otters. Okay, so even though it's not affecting them directly, because they're not obviously breathing the oxygen in the water, they can come out on land and they can breathe themselves. Okay, it is obviously having an impact. Okay, so what we've got is we've got a decrease in oxygen concentrations in the water, which will cause a decrease in fish, because the fish can't jump out of the water and breathe the air. And that is then because fish need oxygen to respire. Explain. So the otter population decreases due to lack of food. Okay, kind of makes sense, really, doesn't it? Question number 6. Hormones. Nothing like a good hormone. Hormones are released from endocrine glands. That's correct. Which gland releases hormones to control other glands? Okay, so let's think about it. What's the big gland? The master gland. Okay, so it's not adrenal, okay, because that is just adrenaline. Pituitary is the master gland, and then the thyroid then is in your neck, and that controls metabolism. Several hormones can affect the blood glucose concentration. Okay, so there's really kind of two. If your blood glucose levels go up and your blood glucose levels go down. Adrenaline can increase blood glucose concentrations. What is one other effect of adrenaline? Okay, right, so if you think a tiger walks in the room, your blood glucose is going to go up because you need more respiration. Decreased breathing rate? No, unless you're dead. Decreased metabolism rate? No, that's probably after the tiger's eaten you. Increased blood flow to the muscles? Yes, because you need to run away. Increase FSH. So do you really want to produce more eggs in the ovary if a tiger's running at you? So it's definitely not that one. 6.3. Okay. Cells in the pancreas detect changes in blood glucose concentration. What type of cell in the body detects change? They are receptor cells. Okay, and I'll often call them that you've got the alpha and you've got the beta cells in there, but they are generally receptor cells. Or sometimes you can call them sensory cells. Scientists investigated whether blood glucose concentrations affect reaction time. Okay, so if you've got a load of sugar, do you react faster or slower, or however it is? The reaction times of 10 people with type 1 diabetes, so type 1 diabetes is the one you're born with, right, where you're not basically producing enough insulin. This is the method used. Tell one person to drink a glucose drink. Yum, yum. Monitor the person's blood glucose concentrations, taking small samples of blood. Record the person's reaction time when the person's blood glucose concentration is at those 3 individual places. Repeat steps 1 to 3 for the other 9 people. Okay, this is your ruler drop practical. Okay, so remember you have your left hand or usually your weaker hand on the end of a table. You have your thumb and forefinger 2 centimeters apart. Your partner then drops the ruler in between your fingers while you're looking away. Okay, and then what you do then is you've got your centimeters on the ruler, use a conversion table, which gets you to your reaction time. People with type 1 diabetes were selected for the investigation instead of people who did not have diabetes. Why? Okay, so that is then because blood glucose concentrations, can't spell that at all today, varies more than in non-diabetics. Okay, so that means no insulin is produced when glucose increases. So basically what it's saying there is that you kind of stay away from the normal homeostasis response or position for longer. So then what you can do is you can do the test for better. 6.5. Control variables between different people in the investigation include age, sex, food and drink consumed before and during the test. Okay, so everything's kind of kept the same. So same sort of age, same sort of sex. Okay, there's only a couple of choices. Food and drink consumed before and during the test. Suggest 2 other control variables that should be used in the investigation. Right? It must be the same type of test. Okay, obviously what they're doing on this one, they're talking about the ruler drop. they must control other substances, e.g. caffeine or alcohol. You must have the same amount of sleep. There must be no distractions. Okay, so that's just a few different things that then could be kept the same. 6.6. It was important for the scientists to monitor the health of each person during the investigation. Suggest one reason why. Low or high blood glucose could be dangerous. What it could do, unlikely, it could bring unconsciousness. Oh my God, I'm never going to be able to spell that one. Unconsciousness. You could get into a coma. Much more minor, is you might get a really bad headache from it. Okay, so you've got to be monitored, okay, because if your blood goes down or low or high or whatever it may well be, you could get some sort of diabetic effects. 6.6. Figure 6 shows the results. Okay, people who do not have diabetes usually have a blood glucose concentration in the range of 4 to 7.8. Okay, 4, which is around about there, to 7.8, which is around about there. Describe how the results in figure 6 show the importance of homeostasis. So the lowest, so if you look at it, that's the lowest there. Okay, that's the lowest mean. All right, the lowest reaction times are recorded for people without diabetes, okay, which is this section here. And that's then because homeostasis keeps blood glucose levels in a narrow range, which is the optimum. 6.8. Describe how blood glucose concentration is maintained with narrow limits in people who do not have diabetes. Right. Okay, this is a good one. Let's change colour. What colour should we have? Let's have red. Okay. So, first one, maintained in narrow limits. So what we could do is we've got to talk here about blood glucose going up and going up down. So blood glucose increases. So this is in people that don't have diabetes. Blood glucose increases. The pancreas releases insulin. Okay, and then what insulin does, insulin causes glucose to be converted into glycogen in the liver. Okay, so basically what it is, it's taken out of the blood. Okay, let's do it a different colour so it's obvious. Okay, oh, that's still red. And it's still red, make it go purple. Blood glucose decreases. So therefore, the pancreas releases glucagon. And what glucagon does is glucagon converts glycogen into glucose. Stupid loads of G words. Okay, so glucagon's a hormone. Glycogen is the store of glucose in the liver. Into glucose, glucagon converts glycogen into glucose in the liver. So then what? Blood glucose increases. Increases. 6.9. The scientist gave the hypothesis optimum blood glucose concentration increases the production of chemicals that diffuse across the synapse. Okay, so we've got a synapse. There's a synapse. Oh, that's terribly done. Synapse. Okay, so the synapse is that little gap there, the synaptic cleft. Describe how an increase in the chemicals could decrease reaction time. All right, so if you've then got more chemicals getting released into the synapse, okay, how an increase in the chemicals could decrease reaction times. Right, so if more chemicals diffuse across the synapse, an impulse is more likely. So, more likely for muscles to contract. Okay, so therefore, if more chemicals cross, they diffuse across, an impulse carrying on down is then more likely, and if it's more likely for the muscle to contract, reaction time, so reaction time decreases. Okay, that's paper 2 higher completed, right? Make sure you're studying it, make sure you think about exactly what you're doing. Okay, so it's definitely a case of good luck, okay, in your paper itself.